So I would expect you to be able to draw out kind of a map of everything we've talked about in this chapter. That will show you that you are really starting to understand it. But let's start here. I'm just going to draw out some of the stuff we've talked about recently, because I think the ETC is one of the harder parts to really grasp. So we have NADH and FADH2. And now I want you to tell me where these came from. First of all, where did those come from? You should be able to tell me that. I'm not going to tell you. You can rewatch the video. But you need to know that, right? Where did those come from? How are they made? What are they? What do they contain? Now, there's an electron transport chain that consists, so there's a membrane. I'm not going to draw the membrane, but there are these complexes. There's complex 1, 2, and I'm drawing them lower because as we go down, it means they're losing energy, right? The electrons are losing energy. So there's complex 1, 2, 3, and 4. And NADH dumps electrons off, 2 electrons here. FADH2 dumps them here. The electrons travel down these. And what's waiting at the very end? Oxygen, right? This is why you breathe. You exhale CO2 that we talked about in other parts. So CO2 produced during the oxidation of pyruvate, and CO2 produced in the citric acid cycle. That's why you exhale. You inhale to get this oxygen for this reason, to run cellular respiration. I'm going to ask you that on a test. Why do you breathe? Or why do you need oxygen? And if you answer to breathe, I'm giving you a 0 on that question. To breathe is a kindergarten answer. I want a scientific answer. It's the last electron acceptor in the electron transport chain, right? So electrons come down. They're drawn towards oxygen. Oxygen's like a big magnet pulling those electrons towards it because it really likes electrons. The energy from those electrons is used to pump hydrogen ions out here. They're pumped like crazy out here. And remember, hydrogen ions were a product of several of the reactions we looked at earlier. And not all of these are hydrogen pumps, but I drew hydrogen pumps all over the place. So we have hydrogen ions in high concentration out here, which is the intermembrane space. I'm just going to put intermembrane space, IMS. And down here is the matrix. So there's actually a membrane, right here that all these things are stuck in. Now, there is one more thing here, and that is this kind of tunnel right here. I'll draw it kind of like that. This is ATP synthase. These hydrogen ions can flow back through this way, back into this side. And by doing that actually ATP synthase spins. This makes kinetic energy, and that kinetic energy allows it to grab a hold of ADP and phosphate molecules, and allows it to make ATP. And ATP, that molecule we've talked about before, I've drawn it before, it's got a 5-carbon sugar, which I'll screw up there, and that's fine. Adenosine on one side and 3 phosphate groups. This is incredibly quickly and crudely drawn. The sugar is actually more like this, right? I just kind of screwed it up. Anyway, it makes that. It sticks the phosphate groups on there, right? That's how we power it. We go from, notice, we go from potential energy. All these hydrogen ions up here, this high concentration of hydrogen ions is potential energy. As they flow through, it's kinetic energy. And then we convert that back to the potential energy of ATP. So you think about that. We pump hydrogen ions to one side of a membrane. So all that high concentration on one side is potential energy. They flow through to the other side. That's kinetic energy. That kinetic energy is then locked up into potential energy again, the potential energy in the bonds of ATP. Now, what if there was a molecule? We'll call it compound X. Compound X binds to complex 4 and stops electrons from flowing. So if you get a hold of compound X, it actually sticks right here. And it prevents electrons from going any further than this. So oxygen is down here, but it's doing nothing. What would happen? Well, I'll ask you, think about it. What would happen if a molecule stuck right there and prevented electrons from flowing? Well, ATP would no longer be made. oxygen would not be utilized. You would essentially suffocate even though you were breathing. And there is a molecule that does this, right? So pretend this is pre-social distancing. Pretend there's a big line of, electrons flowing through the transport chain. What if someone was blocking the flow? They can't move. They can't get through. That would lead to the stopping of ATP and death. And there is a molecule that does that. There is a molecule that stops electrons from flowing. It's called cyanide. So that should give you some idea of how important this process is. You get a little bit of cyanide in your system and you're dead in a few minutes typically, because ATP needs to be made constantly to keep you alive. So the only path for hydrogen ions to get back into the matrix is ATP synthase. And ATP synthase actually spins. And you can see some animations of this in your study tools on mastering. It spins, and that spinning, that kinetic energy gives this the ability to grab ADP and a phosphate group and to glue them together to make ATP. Kind of like water flowing through a dam can be used to generate electricity. Okay, so I want to point out that this same process is going to be used in chloroplasts, except the parts are going to have different names. So this is a great figure to study later. Mitochondrial structure, you can see here, hydrogen ions are pumped from the matrix into the intermembrane space, and then they flow back into the matrix. There's different terminology for the parts of the chloroplast, but they will use a similar process, which we'll talk about in a future lecture. I just wanted to point that out now because I want you to be ready to understand there is a similar process here in the chloroplast. Now, we have focused on sugars in this lecture, so carbohydrates that go into glycolysis, and then they become pyruvate, and then they become acetyl-CoA, and then they go into the citric acid cycle, and then their electrons go into the process of oxidative phosphorylation. But I want to point out that proteins and fats can also go into this process. The different parts of these molecules will enter in different places, and that's why it's kind of important if you're going to go into medicine or health to understand chemistry. I mean, that's one of the many reasons, right? And so bear that in mind. Like proteins, for example, will enter in different phases. The amino group will actually be peed out as urea. Parts of them will enter in here, here, and here. And fats, this part right here will be burned up if you're not doing aerobic exercise. But if you're not doing aerobic exercises, these big fatty acid tails won't get broken down. So anaerobic exercise, like, I don't know, fencing or weightlifting, is not good for losing weight because you don't burn fat that way. So you can learn a lot by studying this chemistry. But I just wanted to point out that it's not just sugars that are used for food. Other macromolecules are food as well for us. So you can look at this figure now and hopefully make some sense of it. So glycolysis, glucose comes in. It produces 2 pyruvates, and we get 2 ATP. NADHs are also produced. They enter into the mitochondria. I'm not worried about, I don't know why let's not worry about that. It's just confusing. Okay. All right. So these pyruvates come into the mitochondria. That's where they're oxidized to acetyl coenzyme A. So for these 2 pyruvates, we would end up with 2 acetyl CoAs. Then we're going to get more NADHs. So in this graph, we're following a whole glucose the entire way, right? So these acetyl-CoAs go into the citric acid cycle, where we would get 2 total ATP out of that entire glucose. We're going to get more electron shuttles here, 6 NADHs and 2 FADH2s, which are all of these electrons end up going into the electron transport chain. And we're going to get about 26 to 28 or so ATP out of that. And total, we're going to get 30 to 32 ATP out of this whole process. When you add up these 2, which are made by substrate level phosphorylation, and these here made by oxidative phosphorylation. I do want to point out that bacteria are a little bit more efficient than we are at this, because bacteria do not have FADH2. They only have NADH. So NADH dumps its electrons off partway down the chain, so they have less time to lose all their energy before they get to the bottom. So they're going to generate more heat that's just wasted into space I don't know why we evolved that, but we have it, and it is a bit of a waste. It's kind of interesting that happens. Another thing that I'll mention before I go, just because it's really cool, is and you can research this to learn more about it than I know, because I don't know a lot. But in babies, they have something called brown fat, and brown fat has a ton of mitochondria in it. But the mitochondria often, I think they have membranes that have holes in them, or they don't have much ATP synthase. So they produce a bunch of these hydrogen ions that just flow back through and don't accomplish anything. But the process of running the transport chain generates a lot of heat, because when you transfer energy, you generate heat, and it allows babies to generate heat and stay warm without producing excessive ATP. Kind of cool. Anyway, so let's see, where are we at? So in the next video, I'm going to talk about what happens if you don't have enough oxygen around to do aerobic respiration. You don't have enough oxygen at the bottom of that transport chain to pull those electrons down. What happens then? And that's where we'll go, and we'll talk about how to make beer in the next video.